the stomach has had its way with the chyme. It's introduced hydrochloric acid to it, it's introduced pepsin to it, it's introduced several other things into that mixture to help break down that food that you've eaten. Hydrochloric acid is really good at breaking down protein along with pepsin, which does a great job at breaking down tissues as well as lysing and breaking open cells because, well, you got a lot of proteins in that cell membrane that, if they're broken, would cause the leaking of its contents out into the stomach. Now we're going to move on to the small intestine. And to be honest, you're not ready, that food isn't quite ready to be absorbed yet. There's still quite a few other issues going on that you got to overcome. First of all, yeah, that pepsin has broken down proteins into dipeptides, tripeptides. They've broken down that polypeptide into smaller pieces, but you still got saccharides, you still got sugar, carbohydrates, you still have other proteins, you still have nucleic acids and a bunch of fatty acids in the way. You've got to break it down to the carbohydrates down to monosaccharides and disaccharides. You've got to break down those proteins down into amino acids, dipeptides, tripeptides. You got to break nucleic acids down more fully, as well as the lipids need to be broken down into monoglycerides, small fatty acid chains, and things like that. Another issue you've got going on is that you've got to get some of the accessibility to fats. Fats don't like water. And to be straightforward with you, the majority of the stuff that you eat, as well as what is introduced into the small intestine by your own tissue, is hydrophilic. So that fat tends to try to glob together into its own little ball, into its own little clique. And that's not good if you really want to break things down. The more surface area you get, the more you break down. So we've got to overcome all of these different issues in order for our bodies to take up what it needs to be able to survive. So what happens at the very beginning of the small intestine, there's the very first portion the small intestine is called the duodenum. The duodenum is very short, 6 to 12 inches. But there's a lot of activity going on here. There's a lot of insertion points into it. You'll see here's a drawing. We see the liver, gallbladder, and pancreas, all of which has some type of ducts leading into the small intestine. These are called accessory organs. Accessory because they don't have a direct line or contact with this food. But it still provides some means of chemical digestion. The liver producing bile salts. The gallbladder does not, I repeat, does not make bile salts, but it stores it and concentrates it because your liver is constantly making it. And it's secreting it. If you don't need it, it gets sucked up into that gallbladder and then concentrated, which explains why some individuals develop gallstones. It's extremely concentrated bile salts that have solidified and crystallized. The pancreas is responsible for two main things. One, the enzymes that are going to digest those larger molecules down into the simplest of forms of all those different types of nutrients, as well as the things that a buffer, a sodium carbonate, to help buffer out that hydrochloric acid. We no longer want the hydrochloric acid because it's not going to be great for the duodenum. It was there for purification. It was there to make certain that bugs or bacteria that are going to sneak through get knocked out and that things can be emulsified as best as they can. Not emulsified, liquefied as best they can. You will notice that a lot of these, there's some lines or tubes in all of these digestive and these accessory organs. And they do have names, and yes, you do need to know what those names are. I'm going to go ahead and point out the ones that you do need to know. The right hepatic duct and the left hepatic duct, both of which will be collecting bile from, well, you betcha, all the different lobes of the 4 different lobes of the liver and combining it all into the common hepatic duct. HEPA or hepatitis or anything that says HEPA in it refers to that it's part of the liver. So left hepatic duct is just saying the left liver duct. The right hepatic duct is just saying the right liver duct. Common hepatic duct is just saying it's the common liver duct. And so it's where all of these bile ducts have come together into one. Now, depending on your situation, it'll reach this bile will reach this junction point, in which it'll either suck up or pull into up through the cystic duct into the gallbladder, where it'll concentrate that bile. Or if you're actively eating and you need to get that bile down, it's going to go into the common bile duct, which then it will eventually reach the hepatopancreatic ampulla or the ampulla of Vater. Hepatopancreatic is the one that's used most frequently. The pancreas has its own duct. It's called the pancreatic duct. It's a nice and easy name. There are two ducts that are going to sneak out. The accessory duct, or aka the duct of Santorini, just called the accessory duct, or where it binds together with that common bile duct and it just becomes the liver, pancreatic, pancreas, the liver pancreas ampulla, hepatopancreatic ampulla. There it is in real life. I almost said in cartoon form. I'm glad I didn't. Oh, I already did. You will see that you do have those ducts combining together. It does get released out into that small intestine, that duodenum. We are in the retroperitoneal cavity. Now, I had mentioned this a little bit already. I do need to mention how, again, that pancreas releases the enzymes for the digestion of carbohydrates, proteins, fats, and nucleic acids, as well as that sodium bicarb. That way you can buffer out that hydrochloric acid. But the question is, how does it know when to release it? Obviously, you don't want to be spewing out enzymes all the time. It costs energy to make those. It costs resources like amino acids to make those enzymes. And it takes a lot of effort to make sodium bicarb. So we really don't want all that just excreting all the time. How does it know when to notch it up? Well, if you remember, gastrin does its thing. It causes the stomach to open, the pyloric sphincter to open up. It causes hydrochloric acid production. So when that pyloric sphincter opens up, that chyme gets released. Portions of chyme get released into this duodenum. And you got several sensors going on in here in this duodenum. I'm going to go ahead and sneak forward a little bit. Several sensors going on here. The one that we care about is the enteroendocrine cell. This is the one that's going to secrete the two of the hormones that we care about, the secretin and the cholecystokinin. Secretin, so these enteroendocrine cells sense both hydrochloric acid and these different types of nutrients that just came from the stomach. The enteroendocrine cell that is responsible for releasing secretin is sensing hydrochloric acid. Reason why is secretin is responsible for signaling to the pancreas, hey, the stomach just dumped stuff in me. I'm not too happy about this. Can we buffer it out a little bit, make it a little less acidic? Can you secrete stuff for me, that sodium bicarb for me? And this pancreas goes, yeah, sure, why not? And releases that sodium bicarb. Enteroendocrine cells that produce and release cholecystokinin are sensing the nutrients. They're sensing the fatty acids. They're sensing the amino acids, the nucleic acids, the carbohydrates, and it's going, hey, there's some rich resources here. Let's get these things rolling. And cholecystokinin is released, and it comes to the pancreas and says, hey, we got to release this. And it releases those enzymes. It's that the cholecystokinin says, we got materials here that we need to break down. Let's break down these materials, send out those enzymes, and the pancreas obliges. Cholecystokinin also does a couple of other things. We don't want that continued release of gastric content, stomach content back into the duodenum. So cholecystokinin says, okay, that's enough. We don't need any more. It also makes its way up to your hypothalamus and says, hey, we are finally getting food into this small intestine. You can start saying to yourself that we're full. That's why sometimes it takes 10-15 minutes, even though you've engorged yourself on food. You still have that feeling of, oh, I'm so hungry, because sometimes it just takes a little while to get some of that food into that duodenum and getting that cholecystokinin circulating. That cholecystokinin, by the way, also has a little bit of effect on the gallbladder as well as in causing its contraction. Because I have mentioned bile and bile salts several times over now, but I haven't yet told you what these bile salts do. It is responsible, you need to know this word, it is responsible for the emulsification of fats. Emulsifying fats that's a really fancy way of saying that it's making fats go into water a little bit easier. Kind of like what soap does for your hands. You got grease on let's say you're working on a car, you're messing with greasy meats as you're cooking, or maybe, I don't know, something that involves some type of oil. And you need to get rid of that oil. While sticking underwater, you're still going to feel that oily gooiness on your fingers. How do you fix that? Well, you put soap on it. Soap has both hydrophilic, meaning loving water, and hydrophobic, meaning loving fat, or hating water, parts and pieces to it. So that way the hydrophobic portion binds onto the fat while the hydrophilic portion interacts with water, thus pulling away fats from one another and making them more emulsified or more easily dispersed. Hence you can get the lipases to them and make them do their thing, allow them to do their thing a little bit more easily. So bile salts emulsify fats. We do need to talk about the structure of the liver really quick. And because, well, obviously it does have some bile activities to it. It's secreting bile and all that. But there's some other things that it does. If you remember, hopefully back in A&P3, there's the hepatic portal system, in which all the nutrients from your small intestine, large intestine, and portions of your stomach come around and are filled into the liver so it can process and do several things with those nutrients. If it's a sugar, it might store it up as glycogen. It might process drugs. It might process toxins. It might check for any microbes or anything like that. It's got a lot of roles that it's got to do. There are several, how it is organized, the histological organization of this. You're going to see that there is a portal triad as well as the central vein. The central vein is what's taking the blood back out of the liver, but the portal triad has, well, kind of like the name indicates, it has three things going on with it. This drawing has a lot of labeling and things like that, but I just want you to look at these three things. 1, 2, 3. It has what they have here, colored red artery and a colored blue vein, both of which mix their blood together. At this point, I usually ask a question about why you would want two different types of blood flowing, but since I don't really have anyone to bounce this off of, I'm going to go ahead and tell you that you need this blood that's blue, that's bringing in all these different nutrients from the small intestine, most likely have expended all of their oxygen. So that means you need to provide the liver with oxygenated blood as well. And well, the easiest way to do that is, well, is just mixing the two bloods together. That way you can have the nutrients going through as well as the oxygen going through at the same time. The other triad, or the other member of this hepatic portal triad, is this bile duct. And instead of flowing outwards toward the central vein, they are often these cells are often making bile ducts in a separate canal that are eventually going to make it to that left or right hepatic duct, and eventually into the common bile duct, etc., etc. There are several theories behind how to describe the flow of these nutrients and oxygen and all that stuff. The one thing to point out, though, that this honeycomb shape is not present in humans. This is more of a pig liver type of thing. It is a pig liver thing. We don't have that nice honeycomb shape. Only pigs do. What these all are telling us just how it's divided. We can't really use this one as an example because it's saying this is a normal unit, hepatic lobule. This is what it should look like. You're surrounded by 3 portal triads to 1 central vein, getting the exchange of blood going this way with the bile salts getting excreted down into that bile duct. But because we don't have that pretty nice setup like we do, it's not really referred to very much as a hepatic lobule. The most of the time we talk about is this hepatic acinus, which is you have this hepatic portal system. Most likely what's going to happen is that blood or what have you is just going to try and find a direct line to that central vein, whether it be this one over here or this one over here. It doesn't matter, as long as whichever way that blood pumps through, it's going to go that way. Finally, let's look at this part E. This is the description of the hepatic acinus. You will see that it is broken down into 3 different zones. And the best way to describe it is to pretend that you are one of these cells in a liver. You're a liver cell, and you are sentient. You are able to think and decide, hey, I want to live in this particular region of the liver. I want to be the best suited and the happiest I can be. Which of these 3 zones do I want? Well, your realtor shows you zone 3. It looks decent. It looks okay, but meh. I kind of want these nutrients, you tell your real estate agent. I'm kind of worried that these nutrients are really far away from me. I might get sloppy seconds or thirds from zone 1 and zone 2 residents. Let's check out zone 1. So you get zone 1. It has a nice, rich wonderfulness of oxygen and nutrients and processing. But then, for some odd reason, the host decides to take a big, giant swig of something. Lots and lots and lots of alcohol. And you see all these toxins flowing through, and it's not doing so well to you and your neighbors, and you're like, that's no bueno. So problem is with being in zone 1 is that although you're getting all the nutrients right off the bat, you're also seeing the toxins right off the bat. Another bonus is if the host body is, I don't know if I should refer to it as a host body, but I'm going to go ahead and do so. It's probably not perfect terminology, but if the person stops breathing or something like that, these cells in zone 1 are less likely to die. They're the last to die in the liver. The ones in zone 3 are most likely going to die because, well, they're most likely going to they won't get to see the sloppy seconds or even the sloppy thirds, because they're going to get eaten up. But the bonus of living in zone 3 is, well, if there's a whole bunch of toxins hitting the body, these guys are going to hit the highest concentration, while you're going to see the lower concentrations right off the bat. Histological cut of both the central vein and the hepatic portal triad. There is the central vein, nice wide open. You got some red blood cells. You can kind of see over here in the middle, you'll see that hepatic artery with its smooth muscle having a constrictive feel to it. We got also the hepatic portal vein being nice, wide and open, nice thin layer like we discussed in AMP3 about all veins. And then we got the bile duct. You can see it's really quite small. You're not producing very much bile at once. One last thing to point out about this bile before I move on to the discussion of other things about the liver is that bile, remember how it's fat soluble. What is going on is that things that you don't want that are lipid soluble in your body, like let's say you have too much, males, you have too much testosterone, too much cholesterol. Females, you might have too much estrogen, and they're trying to cycle through things and get things moving, going on. How do you get rid of it? Well, you can't pee it out because it's hydrophobic. These steroids are hydrophobic. You got to excrete it out via bile, and that's exactly what you do. So when you secrete bile, you're also secreting that fat-soluble waste products. A lot of other things that the liver does. It does process that carbohydrate, lipid, and protein. It has a lot to do, tons to do with drugs and hormones. Like I already mentioned, it's the process of steroid hormones. It's also, if you think about it, that it's the first place that you're going to see a lot of drugs go through. You're going to, pain medicines are modified here, alcohol is modified here, and to be frank, the liver is such an amazing place. And when we talk about metabolism, and enzyme activity, and all that stuff, this is what you're gonna. When we talk about it in metabolism, you're gonna really appreciate that liver. We talked about bilirubin last session. Remember that bilirubin is remnants of the red blood cells being excreted out. It does store nutrients, especially glycogen. That's the way that we store sugar. Phagocytosis, if you got some bad red blood cells, or maybe you got some bacteria that shouldn't be there, it gets phagocytized, as well as vitamin D activation. You get the sunlight, hit the vitamin D, that vitamin D heads back to the liver, where the liver says, oh, you hit sunlight, let's go ahead and flick the switch and you're good to go, and it becomes active. The liver, the gallbladder, one last reminder, it does not make bile. It does not do any of that stuff. What it does do is whatever it stores up, it condenses it and then squirts it out when you need it. Because really the liver by itself, in trying to produce bile, if you had a cholecystectomy, meaning that you got your gallbladder removed, you have been instructed to no longer eat very heavy fat-related items or to take bile supplements. Because without it, the liver can crank out a little bit of bile for you, but not enough, and eventually it does cause the stomach issues, stomach pains. I shouldn't say stomach, but abdominal pains, as well as greasy fecal material. With that, we have discussed in good and decent detail. One last thing really quick about the pancreas, I lied. These particular enzymes, I wanted to point these out before we move on about the discussion about the small intestine and all that. These enzymes we discussed earlier in another earlier video, but I'll go ahead and discuss these here. How to differentiate an enzyme. Most of the time you will find an enzyme, the enzyme's a protein by the way, it will end with an ACE. 90% of the time an enzyme will end with ACE on it. And often the time, whatever it digests or wherever it's from will be in its name. Salivary amylase, remember that amy means sweet. This is coming from, it's in the saliva and it digests sweet things. Lingual lipase comes from underneath the salivary gland, the lingual salivary gland, and it's released under there and it digests fats. A lipid digests lipids. Get into the stomach. Pepsin we talked about, it's one of those rarities where it doesn't have an ACE at the end of it, but we do know that it does break down proteins. Gastric lipase is coming from the stomach and is digested and it digests lipids. It doesn't do it in the stomach because it's too acidic, but once it makes it to the small intestine and that sodium bicarb is introduced, it becomes active. We get down to the pancreas. We'll see tons of enzymes released by that pancreas. Things that will break down every single type of nutrient. Pancreatic amylase. We learned from salivary amylase that ame means sweet. So this has come from the pancreas and it breaks down sweet things. Elastase breaks down elastin. So when you eat that connective tissue, those tendons and things like that, this elastase will be present and will help digest those elastic fibers. Carboxypeptidase begins the digestion of peptides at the carboxyl end. I don't know if we've really talked about that, but it's very particular as to how it breaks down protein. Pancreatic lipase, lipids are broken down. Nucleases, ribonuclease, take a stab in the dark what that does. It breaks down, well, ribonucleic acid. This one breaks down deoxyribonucleic acid. The one curveball that but you will see in science, especially if you're doing cell culture, is trypsin. Trypsin breaks down proteins. I used to use it for my studies when I wanted to see if I wanted to detach cells from the sidewalls of my plate. I would introduce just a tiny bit of trypsin to help break down those binding proteins so I can get those cells detached from that cell. In the small intestine, you do have brush border enzymes, meaning that you have enzymes attached to the outer walls of these membranes. You will notice that they are very specific. Maltase, there is a sugar called maltose that will break things down into glucose. Sucrase will break down sucrose into glucose and fructose. Lactase breaks down lactose into glucose and galactose. You're getting the idea, I hope. With that, I'm thinking that I'm going to go ahead and discuss the small intestine in more of an entirety in a different video. Thank you very much, and remember, you got this.